Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. Today we're plunging into something really counterintuitive. It's a new finding just out in nature that flips a lot of ideas about cancer on their head. Imagine this. Cancer cells, right? They're notorious survivors. Well, it turns out they might be getting this huge energy boost from, believe it or not, our own nerve cells. And we're not just talking signals here. We're talking a literal handoff. Neurons donating their mitochondria, their power factories, directly to cancer cells, fueling their spread. It's wild. So today we're going deep into this paper, unpacking how it really shifts our view of metastasis. Let's get you up to speed.
Yeah, it's fascinating because it connects things we've sort of known for a while, but in a completely new way. It's like finding a hidden superhighway.
Okay, let's unpack that. Before this paper, what did we actually know about nerves and cancer? Was this link even really solid?
Oh, absolutely. The link was definitely there. We've known for ages that tumors aren't isolated islands. They actually get heavily innervated. Lots of nerves grow right into them. And the data, epidemiological data, looking at large patient groups, consistently showed this correlation. More nerves in a tumor often meant, well, worse outcomes. More aggressive, more likely to spread.
But the why was fuzzy.
Exactly. The why was mostly pinned on signaling. The idea was that neurotransmitters, the chemicals nerves use to talk, were somehow pushing the cancer cells to grow or move. It was all thought to be about communication, not direct transfer of parts. But here's where it gets interesting. Biologists had seen mitochondria being exchanged elsewhere.
Oh, like where?
Like between immune cells and, say, stressed neurons, almost like a cellular support system. So that sparked this really provocative question. Could neurons be doing the same thing, but, for cancer cells, actually donating their power packs? And critically, would that donation make the cancer better at metastasizing? That's what this study tackled head on.
Right. So, first step. Do cancer cells even need the nerves for energy? How did they test that?
Well, they started with models, breast cancer models,
Yeah.
and they essentially performed a chemical denervation.
Meaning they got rid of the nerves.
Or inhibited them, yeah.
Yeah.
Cut off the nerve supply to the tumor, and the result was pretty immediate, a significant drop in tumor growth. Not just a little bit, a major slowdown. And more revealingly, when they looked inside the cancer cells, they saw this widespread downregulation of metabolism.
Metabolism, like their energy production.
Precisely.
Yeah.
Especially the TCA cycle, the tricarboxylic acid cycle. That's like the central engine for cellular energy. It was sputtering. So that clearly showed, yes, there's a metabolic dependency. The cancer cells were relying on those nerves for fuel, not just growth signals.
Okay, dependency established, but how? How are the nerves providing this fuel? Still signals or something else?
Something much, much more direct. They did these in vitro experiments, growing neurons and cancer cells together in a dish. And they saw that the neurons, when they sense the cancer cells, actually change. They differentiate and form these really close physical connections.
Like touching.
Physically touching, yes. And the cancer cells grown with these neurons. Their mitochondrial respiration went way up, a massive boost in energy production. They were clearly getting something substantial from the neurons.
Okay, so how did they prove it was the mitochondria themselves moving?
That's where the cool imaging comes in. They labeled the mitochondria inside the neurons with fluorescent markers, bright tags you can see under a microscope. And then they watched. And they literally saw these labeled mitochondria moving from the neuron into the cancer cell.
Whoa, how?
Through these incredible structures called tunneling nanotubes.
Tunneling nanotubes sounds like sci-fi.
It kind of is. Think of them as tiny biological pipelines, direct conduits between the cells, wide enough for organelles like mitochondria to pass through. They used fancy microscopy, flow cytometry to count the cells receiving them, even 3D reconstruction to visualize it all. And interestingly, neurons were much better at this transfer than other cell types they tested. They seem particularly prone to donating.
Okay, so mitochondria are definitely moving across, but are they actually working once they get inside the cancer cell? Or just cellular junk?
Crucial question. And they had a really clever way to test functionality. They use something called row zero cancer cells.
Row zero.
Yeah, O0. These are cells they engineer in the lab to completely lack their own mitochondrial DNA,
Meaning,
meaning they can't build functional mitochondria. They can't do oxidative phosphorylation, OXPHOS. Basically, their main power generation system is offline. They can barely survive, let alone grow.
okay, so energy-starved cells.
Exactly. Then they put these row zero cancer cells together with normal neurons. And remarkably, these row zero cells started acquiring mitochondrial DNA from the neurons. Their mitochondria started looking normal again, structurally.
Yeah.
And boom, they regained full OXPHOS capacity. They could make energy properly again and start growing independently.
Wow. So the transferred mitochondria weren't just present, they were fully functional, integrated, and rescued the cancer cells.
Unequivocally. It proved the transferred power packs were the real deal.
That's incredible in a dish. But does this happen in a real living body in vivo?
Yes. That was the next critical step. They looked at human prostate cancer samples first. What they found was that cancer cells physically closer to nerves inside the tumor had a significantly higher load of mitochondria, more power packs near the nerve supply.
Suggestive, but not proof of transfer.
Right, suggestive. So they also looked at patients who were getting chemical denervation for prostate cancer, where they treat one side to reduce nerves. And on the denervated side, the cancer cells had fewer mitochondria compared to the untreated side in the same patient. Stronger evidence.
Okay, getting closer.
But for the definitive proof of direct transfer in vivo, they used mice. They genetically labeled the mitochondria specifically in the mouse's own neurons that supply the mammary gland. Made them glow, essentially.
Host neurons, good.
Then they introduced breast cancer cells. And later, they looked inside those cancer cells. And they found the glowing mitochondria from the host neurons inside the cancer cells. Direct visual proof of transfer in a living animal. They even confirmed it genetically by finding specific mitochondrial DNA markers from the host mouse within the cancer cells. It was ironclad.
And denervation stopped this.
Denervation significantly reduced the transfer, yeah, proving the nerves are essential for this handoff to happen in the body.
This is mind-blowing. Cancer cells stealing power packs from nerves. So how do they figure out what happens to those specific cells that get the boost? Tracking them seems incredibly hard.
Ah, this is where the real technical wizardry comes in. They developed a tool called MitoTracer. This was a major innovation in the study.
MitoTracer, what does it do?
It's a genetic reporter system. Think of it like a permanent tag that gets activated only in the cancer cells that receive mitochondria from the neurons.
How does that work?
Okay, so the donor neurons are engineered to make an enzyme, Cre recombinase, but it's targeted to their mitochondria. The recipient cancer cells have a fluorescent switch gene, normally red, but if Cre gets into their nucleus, it flips the switch permanently to green.
So when a mitochondrion moves from neuron to cancer cell.
Exactly. The Cre enzyme gets released from the transferred mitochondria inside the cancer cell, travels to the nucleus, flips the switch, and bam, that cancer cell and all its future descendants turn green permanently.
That's brilliant. So you can actually follow the lineage of the cells that got the mitochondrial boost.
Precisely. It allows for clear, long-term tracking. You know exactly which cells receive the goods. It moves beyond just seeing the transfer to understanding its consequences.
So what did MitoTracer show? What happened to those green mitochondria recipient cells? Did they become like supervillains?
The results were pretty dramatic, yeah. These green cells, the ones that got the neuronal mitochondria, showed a big jump in stemness potential.
Stemness.
Meaning they act more like cancer stem cells. They were much better at forming new tumors from just a few cells, better at self-renewing. These are hallmarks of really aggressive, hard-to-treat cancer cells. They saw this clearly in tests, like sear formation assays.
And metabolically, did they confirm the energy boost?
Oh, yeah. Enhanced oxidative phosphorylation, higher ATP production, basically. They became energy-generating machines, much more aerobic. They also handled stress better, improved redox balance, making them tougher against things like free radicals.
Okay, so more energy, more stem-like. What about surviving the journey of metastasis?
That too. Critically, they became significantly more resistant to stressors encountered during metastasis. Think about oxidative stress, but also shear stress.
Shear stress, like physical force.
Exactly, the physical force and turbulent cells experience when they're tumbling through the bloodstream. These green cells were just tougher, more resilient, built to survive that brutal journey.
So they're super resilient, pre-armed for metastasis. Did that translate to actually spreading more effectively?
Well, interestingly, in a simple dish test, they weren't necessarily better at invading locally, but the in vivo results in actual mice were clear. These mitochondria recipient green cells had a significantly higher metastatic potential. They were much more successful at leaving the primary tumor and setting up shop in distant organs.
And this matched what they saw in human samples.
It did. They looked again at human breast cancer samples and found that cells in metastatic sites consistently had a higher mitochondrial load than cells back in the primary tumor. It all pointed the same way. Getting these neuronal mitochondria gives cancer cells a real edge for spreading.
It really paints this picture of metabolic theft enabling the whole metastatic cascade. How did they finally confirm these supercells are the ones making it to distant sites?
That's where the lineage tracing power of MitoTracer really shone. They used it during actual metastasis experiments. They transplanted little tumor spheroids containing both the MitoTracer nerve cells and the cancer cells into mice. Let the tumors grow.
And then looked for the green cells.
Exactly. They collected cancer cells from the primary tumor and then also from places where it had spread, like the lungs and, significantly, the brain. And here's the kicker. The percentage of green cells, the ones that had received neuronal mitochondria back in the primary tumor, was way higher in the metastases compared to the primary tumor.
So they were enriched. Selected for.
Massively enriched. In the brain metastases, for instance, sometimes up to nearly half the cancer cells were green. It showed that acquiring those mitochondria early on dramatically increased the odds of that cell successfully founding a distant metastasis. It wasn't random.
And this wasn't just one cancer type.
No, they validated it further. Labeled endogenous nerves in mice, tested melanoma too. Saw similar patterns, particularly that strong enrichment in brain metastasis.
Why the brain specifically? Is there something special happening there?
That's a really good question. And the thinking is, yes, neurons themselves have incredibly efficient mitochondria because, well, the brain needs a ton of energy. And the brain environment itself is actually quite tough for cancer cells metabolically, relatively nutrient poor compared to other places.
So getting these super efficient neuronal mitochondria is like getting the perfect toolkit for that specific environment.
Exactly. It might give those cancer cells the precise metabolic advantage they need to not just survive, but actually thrive and grow in the challenging brain microenvironment. It's like natural selection at the cellular level, favoring the cells that stole the best power packs for that location.
This really does feel like more than just an interesting finding. It feels like, as you said, a paradigm shift. What are the biggest innovations here, and where does this lead us?
Well, it firmly puts mitochondrial transfer on the map as a key driver of metastasis, not just a weird side observation. That's huge. And technically, MitoTracer is the standout innovation. Being able to prove causality in vivo, to link the transfer directly to the metastatic outcome via lineage tracing, that's something older dye methods just couldn't do reliably. It's a powerful tool for the field.
And therapeutically, what does this open up?
It opens up completely new avenues, these neuron cancer organelle highways, these tunneling nanotubes. They become potential drug targets. The need is urgent, especially for cancers like triple-negative breast cancer that spread aggressively. If you could block this metabolic support, cut off this energy supply line from the nerves,
You might be able to stop metastasis before it really takes hold.
that's the hope. It's a totally different angle than just trying to kill dividing cells. Could be a powerful strategy to curb that lethal spread.
But I guess the big question is, can you do that safely without hurting the nerves themselves?
That's the critical question for future research, absolutely. Can we block this transfer without causing neurological side effects? We need to understand the exact molecular mechanisms, the specific proteins involved in forming those nanotubes and mediating the transfer, the handshake, so to speak. And another question is, why neuronal mitochondria? Is it just because neurons transfer them more often, or are the mitochondria themselves inherently better, more efficient? Understanding that could lead to smarter therapies.
Lots to explore still.
Definitely. This work throws open the doors. We need to dig into the mechanisms and figure out how to safely disrupt this process.
So the next time you think about how cancer spreads, it's not just genes mutating or cells growing wildly. You have to picture this almost sinister act of theft happening too. Cancer cells literally hijacking the energy factories from our own nerves to fuel their journey. It's a complex and frankly quite unsettling interaction within our bodies.
It really is. And it makes you wonder, right, if we can map out that specific molecular interaction, that handshake allowing the transfer, could we design a drug that just blocks that? something that stops the donation without harming the donor nerve cell or the recipient cell in other ways, preventing metastasis without broad toxicity.
Hmm.
That's the ultimate goal here, a
Yeah.
fascinating challenge.
A truly deep dive revealing how even our own nervous system can be exploited by cancer, and how science is uncovering these hidden pathways, offering new hope.